Hi there. Welcome to session 3 of microbiology and immunology. Let's get the slideshow going. One sec. Oh, I'm not sure the video's loaded. Okay, that's fine. It doesn't matter. You don't really need to see me. You just need to see the slides. Right, let me get the slideshow going. Okay. So today's session, final session on microbiology and immunology, and we are going to be looking at some diseases. So what the assessment criteria says is you need to, or criterion, it's just one, you need to identify examples of pathogenic viruses, bacteria, fungi, protozoans, and worms. Essentially, that's what those helminths are, and explain how they cause disease. So we're going to have a look at some of my favorites. I have favorites because this is my topic that I teach mostly. This is the one that I used to teach at Cardiff University. So I've got lots of favorite pathogens. Um, so what is a pathogen? It is a disease causing bacteria, viruses. Viruses are always pathogenic, fungi, protozoa, etc. So pathogenic means disease causing. Got loads of bacteria that lives on us that's really helpful. We've got loads in our gut, for example. So when we're talking about pathogens, we're invariably talking about disease, and diseases have a specific course. So first thing is infection. Last session, we were talking about infection because we were talking about immunity, and we were talking about all the ways we try and avoid infection, the barriers, the immune system, etc. But once we have been infected, let me just close this little box. There is an incubation period of the disease, and this is the time between infection and when the symptoms appear. The length of this is variable. It depends on the disease. Sometimes it can be up to 2 weeks when you can be actually incubating the disease. It can be quite a long time with some pathogens. This is often the phase where we're waiting for our immune system to kick in properly and start to identify what we've become infected with. If we've been vaccinated, this often doesn't even happen. This is when we can nip it in the bud and stop it progressing into full-on infection. So during this incubation period, the person may or may not be infectious. They often are. So it's not great because you don't know you're infected and you're going around spreading the pathogen to lots of people who you're in contact with. Again, it's very variable, depends on the disease. And during incubation, the thing that's multiplying, the virus, the bacteria, the protozoa, they will reach the number necessary to produce symptoms in the host. And then the immune system works hard to contain the infection during this period. But as we all saw during the SARS-CoV-2 pandemic, the COVID pandemic, it can happen that you carry a disease asymptomatically, which you can spread to other people. And in other people, it can present very differently because we've all got extremely different immune systems that have had different experiences with different pathogens and will respond differently depending on our health. So, a symptom of a disease is something which is experienced by the patient, often pain, fatigue. Well, what happens when you get ill with, you know, lots of different symptoms, coughing, sneezing, difficulty breathing. It very much depends on the disease itself. Symptoms are variable, very variable, depending on the disease. But a sign of disease is something which can be measured. So symptoms and signs are slightly different. So symptoms are something which is experienced and a sign is measurable, such as a high temperature or a high blood pressure or a loss in weight, for example. So those are causes of disease. The course of disease, not causes. Infection, incubation, multiplying of the organism during incubation, then you get symptoms and signs that show that you're infected. So to get a disease, it has to be transmitted to you. And there are many different ways that this can happen. And we talked quite a bit about these indirectly when we were talking about our defenses from this happening. So how we defend against inhaling pathogens. Remember that mucus in our respiratory tract, how we defend against eating pathogens, that hydrochloric acid. There's lots of ways. So one route of transmission is direct contact. So this is how infections are spread when you come into direct contact with the organism, when it's left on surfaces. So a really good example of this is athlete's foot, which you pick up from swimming pool floors, verrucas, which you pick up from swimming pool floors, wet floors, or if you're doing any barefoot sports like martial arts or gymnastics in a gym that lots of people use, you're quite at risk from those sort of diseases. Ringworm is another one. Not very pleasant. You know, skin to skin contact will transfer that. There was an outbreak of ringworm at my son's jiu-jitsu gym recently, so he's been avoiding that like the plague. Not quite the plague, but because there's ringworm there. So yeah, not very pleasant. Diseases, as we are all very familiar, can be airborne. This is such an easy route into our systems, as we saw with COVID, but also flu. And no matter how bad that COVID pandemic was, a potential bad flu pandemic could be an awful lot worse. So many diseases are inhaled through the respiratory system, colds, flus, SARS. So SARS-CoV-2, COVID. There was another SARS. Well, I don't know how old you guys are, but you probably don't remember it if you're sort of in your 20s or 30s, maybe, because it was probably 20 odd years ago now. But there was a SARS outbreak that went spread from Hong Kong over to Canada, and all the scientists at the time were looking at that going, uh-oh, here we go. Is this disease X? Disease X is the name scientists give to the disease that is going to cause the next absolutely catastrophic pandemic that overwhelms all of our healthcare systems and kills a lot of people. So when that initial SARS-1 virus, we just call it SARS-1 now because there's been a SARS-2, when that outbreak happened, everyone was looking at it going, uh-oh, but we contained it. And so we sort of calmed down, we relaxed our SARS preparedness. We were all waiting for this really bad flu. We're still waiting for this really bad flu because it looks like we're on the verge of one, but I'll explain about that later. So airborne is a big deal because that's the way you get from one person to millions and billions really, really quickly. So these are the big problems, the airborne ones. Eating, this is kind of like fairly local usually. Can be a big problem if you've had disasters such as earthquakes and water gets contaminated, but it's always kind of local. It doesn't spread globally. Drinking is pretty much the same issue as eating. You're ingesting it, but this time you've got water contaminated rather than food. Food contamination is usually just preparation, to be honest, you know, poor hygiene in the cooking area. Drinking will spread to more people. Or we have had in the UK actually in the past some nasty E. coli outbreaks from butchers killing people, killing not massive amounts of people, like one or two each time, but it's, you know, it's pretty nasty infection of tens of people from an outbreak in a butcher's probably 30 odd years ago. I can remember one happening. Sexual transmission, that has potential to spread quite widely. Look at HIV. That was a new virus, and it started to take hold in the I wouldn't hesitate to say gay community. What we have to say is men who have sex with men community, because not all men who have sex with men identify as being gay, but that took hold in a community of people in the 80s and killed thousands and thousands of predominantly men, but also that has spread through blood infection. People were taking blood transfusions in hospitals that hadn't been screened for the virus, and they were picking up HIV as well. So that's another route of HIV infection. But also shared drug needles could spread it as well. And then this one, animal transmission, is absolutely huge problem because of all of the diseases that are spread by various flies, but mosquitoes being the main one. Um, they spread the malaria parasite, for example, which is extremely dangerous. It kills lots of people a year. So those are our routes of transmission. We'll have a look at them as we look at some of these examples now. So viruses are just quite complex molecules. They're not alive because they don't have cellular structure. And for loads of other reasons, like they can't reproduce without a host cell to live in either. So, well, not live, but to reproduce in. So there's no cellular structure for a start. They don't necessarily have DNA either. Some of them have RNA, which is quite similar to DNA, but it's more of a more simple form. So all living things have DNA. So lots of viruses don't. This is an actual virus, and I know it looks like a robot. This is called a bacteriophage or bacteriophage. This is a virus which infects bacteria. And this structure you're quite familiar with because this is kind of what HIV, flu, and SARS-CoV-2 that cause COVID look like. They come in so many different forms, from helical, icosahedral, so you could get this similar shape to this, but without this structure at the bottom. And compared to your average bacterium, they are about 100th of their size, which makes sense. I mean, this infects bacteria, so it has to be loads smaller than a bacterium to get inside it. So they are really tiny. So they're not living. What they do is they hijack our cells' biochemistry to make our cells make copies of them. So they need a host in order to reproduce. I'm going to show you a video of that in a bit. When they infect a host, the host will eventually become damaged and potentially burst or just release these particles out of the cell. And then they can get into the bloodstream and infect other cells. But they can't reproduce and grow on their own. So they don't survive very well quite often outside of the body. So that's not all viruses, but quite often viruses don't really survive well on surfaces, for example. Not particularly true of COVID, however, and some other viruses. Right, so here is a very well-studied virus, and the reason it is so well-studied is because of its potential to cause pandemic. So global epidemics or pandemics. So an epidemic is like a local outbreak, and a pandemic is when it goes global. So flu virus is renowned for causing pandemics. There have been several. There was one in 1918, which took place immediately after the First World War. It killed millions. It killed more people than the war. The reason there was such a big outbreak after the First World War is because the war had ended. There was thousands and thousands of troops in essentially different countries, all making their way home. So all of this mass movement of people managed to spread the flu virus all around the globe. Then there was another one in 1957, 1967. So these have been big global outbreaks which have killed quite a lot of people across the world. We are on the verge of another one at the moment. You might have heard people talking about a strain of flu called H5N1. That is a flu virus, which is it's a bird flu, an avian flu virus. They're nearly all avian flus, to be honest. The natural reservoir of flu is birds, but we're watching this one very carefully at the moment as well. So it constantly circulates in loads of animals. It moves around in bats, pigs, horses, dogs, and humans. Bats are a massive problem for viruses. Loads of viruses will hang around in bats. So we have to really watch human contact with bats carefully. Reason for that is they are flying mammals, so they have a really good metabolism. They're the only flying mammal. They have a really good metabolism to maintain this high energy state that they need to fly. And they're a mammal, so they're really quite similar to us in their biochemistry and their biology. So that's a problem. They've got all these horrible viruses that they're infected with, but these viruses don't harm them. So they get infected and they just carry on living their best lives. We go and start exploring caves, just recreational caving, whatever we're doing, and end up catching these infections. Or in some places, people will eat wild animals, which um, usually when I tell students that, they're like, what, oh, gross, they're eating bats. But I mean, if you live somewhere rural, certainly when I was growing up, I would often see, um, pheasants and rabbits hanging up outside the village butcher's shop that had been obviously shot by local people. And then they'd just be eaten. So it's not unusual for people to eat the wild animals around, or it's just that in the UK we wouldn't tend to eat bats, but they certainly would get eaten in some communities, and they only have to get into one population of people to spread across the globe very quickly, as we saw with uh, SARS-CoV-2. So that originated in bats. So subtypes of the virus for flu are defined by the surface proteins. You can see these pink and purple proteins sticking out of the virus on the board. There's some that are called hemagglutinin. There's purple on this diagram, and there's some that are called neuraminidase. And then we name the flu virus based on which type of hemagglutinin and which type of neuraminidase they've got. So H5 stands for hemagglutinin number 5, N1 stands for neuraminidase number 1. So that's the one that we're keeping a very, well, I would say we are keeping a very close eye on it globally. Unfortunately, where it seems to be most prevalent at the minute is the United States, where the current administration have rolled back all of the, well, an awful lot of the scientific, not regulation, surveillance, a lot of the scientific surveillance of these emerging diseases, which is extremely worrying. And the scientists over there are concerned, almost panic state. If you speak to them, they are very worried about the lack of observation of this strain. So these different subtypes, so H5N1 is a subtype. It affects how easily it spreads. It affects the severity of the illness in different hosts as well. So at the minute, H5N1, if it gets into humans, is pretty nasty. There's been quite a few deaths, but it's not spreading person to person. That's really important. All of the people who've been infected have been working very closely with animals, so they've caught it off animals. There is a global influenza surveillance and response system which monitors the spread of influenza worldwide, and it informs the development of seasonal and pandemic vaccines. So we get vaccinated every year for flu. If you've got kids in school, or if you were recently in school, you can probably remember being vaccinated. I've got a 15-year-old son. He's been vaccinated this year. They just do the lot of them in school, and that, not that they're particularly vulnerable from flu, but they're the people that really will spread it. Kids really spread disease because of the way they're always in close contact and in confined spaces in classrooms, and then they take it home to older, sicker relatives. So yeah, really good example was that 1918 flu that I've told you about. And the reason we are massively concerned about flu is its pandemic potential. The influenza A subtype H5N1 is shed from the saliva, mucus, and feces of infected birds. Other infected animals can shed it in other bodily fluids, such as their milk, for example. So it's worrying if it gets into cows, such as it has in the United States. The virus can spread really rapidly among poultry flocks because wild birds go, well, wherever, they particularly migratory birds like geese and stuff, they'll go and they'll fly, and then they'll just go and swim in a pond that's got farm birds in. And infect a whole flock of farmed birds. So about 500 million farm birds have been slaughtered in the efforts to control this particular H5N1 strain. It has a really diverse reservoir. It's been found in cows, birds, humans, mink, seal. So it's really concerning the scientists, this one. So It actually says in the notes on the course, which it needs correcting, that humans are the only hosts, but they are not the only hosts at all. This is a really prime example of a zoonotic disease which spreads between animals and people. So I think that's an error in the notes. So humans are not the only hosts and vectors at all. Um, and that therein lies the problem with this disease. It's why new strains keep appearing. So it's spread by airborne droplets from coughs or sneezes. The incubation is about 1 to 4 days. The infectious period, however, is from 1 day before symptoms to 5 to 7 days afterwards. So you can go around infecting people before you're even aware that you've caught it, unfortunately, which is really beneficial for the spread of the virus, works great from the virus's perspective. So it's the type A influenza that causes the most severe diseases and pandemics. It's a very unstable virus, and there's new strains every year due to antigenic drift and antigenic shift. So antigenic drift is when we get small changes, and that gives us usually different strains each year, which is why you have to keep getting a new vaccine every year because there's new strains out each year. And antigenic shift is when you get a major change, a massive jump in the genes, genotype, the DNA, the genetic material. You get a new subtype, you get a big global pandemic. The really at-risk people are babies, elderly, and the chronically ill with this. However, some flu strains have had very nasty effects on young and healthy individuals. The Spanish flu, for example, had a really bad effect on sort of 20, people in their 20s, 30s, teens. That's because they produced a very good immune response, and the secretions of their lungs actually did a lot of damage and killed off a lot of them because their immune response was so strong. So it's not always the people who you would think that are vulnerable are going to be vulnerable. So flu treatment is usually at home, depending on your age and fitness. I mean, older people have to go into hospital because they get so ill, particularly really old, sort of 90s, A plus people. What they normally say is rest, fluids, and antipyretics. So stuff that's going to bring your temperature down, like paracetamol. Stay at home until the fever resolves for 24 hours. To be honest, you're lucky if it's 24 hours. Flu normally lasts quite a long time. People think they've have flu sometimes when they've got quite a nasty cold, or and it's not flu. Flu, you're going to be ill for a while, like a week probably minimum. You can take antivirals, but they don't tend to give them out unless it's high risk case. Antibiotics are sometimes given out, but that's usually because a secondary infection develops. They do not work on the flu itself because it is a virus. So here's the list of symptoms. So they're very generic: headache, fever, achy muscles, achy joints, feeling weak and tired, vomiting, possibly coughing, runny nose, sore throat, and aches in your respiratory system. So they're so generic that people often say they have flu-like symptoms when they get any of these sort of things happening. So that is the flu. The next potential disease X, the one we are really, really worried about, or watching extremely carefully. We have genotypes of all of these viruses, so we know their genome and watch them really carefully. Another virus that has been extremely well studied is HIV. In the 80s, this started to become widespread, as I've said, particularly in men who have sex with men. It spread really rapidly, and there was, unfortunately, really hard to tell if you're infected, because when you first become infected with it, there's either no symptoms or you get a little cold type symptoms. So you wouldn't think it was anything. So people were unaware they were infected, continuing to have unprotected intercourse with other people and spreading it around. So this image shows HIV in green attacking an immune cell. In particular, the virus attacks some white blood cells known as CD4 plus lymphocytes. I'm going to show you some of those in a minute. They are one of the main defensive cells in our immune system, and we need these to activate the B lymphocytes, which give us longer term immunity. Let me just check. Why are viruses able to reproduce if they're not alive? Well, that's getting a little bit into the realms of philosophy rather than biology. Um, if they're not good at surviving outside of a living body, why have they not all died by now? Well, because we'd have to die out to kill them. We are a reservoir of them, and so are all the other
What up,
animals.
Podgy? Why is Podgy even awake?
Sorry?
Oh, nothing. Sorry.
Oh, okay. So yeah, they just exist. We do not know. That's like saying, why do we exist? We don't know. I can't answer that. Sorry. That's not a question for a biologist. That's a whole different subject area. No, it's not a silly question. It's absolutely valid. Yeah, there are ways to prevent HIV now. HIV is considered now a chronic condition. So, like having diabetes or something, there are ways to prevent transmission and infection. So it is really yeah, we've come on a lot since the 80s in terms of what we can do with this virus because a lot of money's gone into studying it. So HIV itself, the virus, doesn't cause an illness particularly. You're okay after being infected with HIV as long as it doesn't overwhelm your immune system. But unfortunately, in the early days of HIV and AIDS, it did overwhelm the immune system, and people developed AIDS, which is acquired immune deficiency syndrome. So their immune system was deficient because of this infection. And that meant that your immune system wasn't doing the right job to monitor and defend against other pathogens. And so your body would become overwhelmed with either those pathogens or cancers because your immune system has a really important role in removing cancerous cells as well. So in the early days of the HIV epidemic, you would end up infected and dead from something really random that wouldn't normally kill people because your immune system couldn't cope with the infection, or you would end up with some strange, unusual cancer that doctors didn't normally see. And they were getting a bit baffled as to why they were seeing these cancers originally. And then they realized, obviously, it was as a result of the HIV infection. So, yeah, it was really horrible. There was a drama series actually on Channel 4 that I can't remember the name of now, but it was absolutely brilliant. But that showed the more sort of social effect on the people that had it at the time because you got horrendously stigmatized for being infected. It's in a way, people used to be historically stigmatized for having leprosy, which is a mycobacterium leprae infection. People were stigmatized because they looked not very good when they were infected with that. So people wouldn't associate with them. And then the same thing with HIV. Um, it was seen as like sinful because it was associated with the men who have sex with men, um, which is really horrible. So yeah, when the virus has started to cause problems and your immune system isn't working, you have AIDS. Now, unfortunately, this is still the outcome for a lot of people in poorer parts of the world. Like I said, in the UK, HIV is just a chronic condition now. You can live with it and you can live a good life and you don't have to infect anyone. And you can live a long, normal life, but obviously with some medical care. But in, unfortunately, poorer countries, you get HIV and it's still most likely going to end up in AIDS and death, which is really, really unfair and horrible. So HIV is transmitted through bodily fluids such as blood or semen or blood products. So you might be aware of the blood scandal that happened in the UK where people were given infected infusions of blood, people with blood disorders. have got it through that route as well. You can just use barrier methods to avoid infection. So using contraception such as condoms, which act as a physical barrier between fluid exchange. It's important not to share needles. There have been needle exchanges in the UK from charities for a long time to stop needle sharing, because you're not going to stop addicts from using drugs very quickly, but you can stop them from getting infected through sharing needles. And we do check our blood products now to prevent transmission of viruses. Right, let me show you how it works at a molecular level. That's HIV. It's a retrovirus. And that's the envelope. That's the RNA. So it hasn't got DNA, it's got RNA. This is a really important enzyme, and it's going to convert that RNA into DNA later. So there we go. We can see the outside of the HIV virus. That is one of our cells. And those little proteins on the surface attach to receptors on our cell. So that's one of our immune cells. You can see these are CD4 plus cells. So that means they've got CD4 receptors. That's what that virus has just attached to. And it's going to use those other little receptors there as well to help anchor it in place. And once it's done that, it will pierce the cell. That's what you can see happening there. And that drags the virus down. And now the membrane on the outside of this virus is going to merge with the membrane of the cell. Remember those lava lamps and that membrane that we looked at all that time ago? This is what's happening. That's merged together like a lava lamp. And now the contents of that virus are being released into our immune cell. So these are proteins surrounding the viral RNA. They just kind of get broken down inside the cell. But there's the viral RNA, the viral genetic information. Now, this little structure is moving towards the nucleus of our cell, and it's got a special enzyme on it called reverse transcriptase. And what that does is it converts the RNA into DNA. So that's one strand of the double helix. Then it will go through another time, do another little twirl through this protein, this enzyme. And just like that, the viral RNA has been converted into DNA. And those little bases there that are building that DNA molecule are ours. They're out of our cell. So it's already using our stuff to build DNA. That's another enzyme. It's called integrase. And that's literally going to integrate it into our cell. So there's our DNA. We're inside the nucleus now. That hole you just saw was a nuclear pore on the nucleus. And there you go. It's just snipped some viral DNA right into our DNA. Nothing we can do to stop it. Well, we've developed drugs that can stop this phase now, but there's nothing in our body that will stop it. So now our body's own protein making machinery is busy starting to make proteins. There you go. That's the nuclear membrane again. Now it's going off to the ribosome. Remember, the ribosomes are the site of protein synthesis. This is on the endoplasmic reticulum just outside the nucleus. And we're building a protein now, a viral protein. Our own cellular machinery is building a viral protein. That then gets packaged, that viral protein, and gets transported to the membrane of our cell. And then it gets moved outside our cell, and it will just sit outside our cell for a while. Now, you might remember these from the surface of the virus initially, these proteins. So there's another little bit of DNA there. Well, it was DNA from the virus, and it's going to make proteins now that are needed to build the virus. So there's another load of proteins that are being made. And they're going to go off to the surface of the cell as well. Remember, this is all happening inside our cell, and they're just going to hang around there on the inside of the surface. And then just like that, that buds off again. Remember those membranes like the lava lamp? And inside, the virus has got all these proteins that our cell has just made for it, just needs to chop them up. Easy job. There's an enzyme for that. And now we've got a fully formed, brand new virus, another one being pinched off there, that can go off and infect a load more cells in our body. As long as they find the cells that have got that CD4 receptor, so our immune cells, they can keep infecting new immune cells. So they can overwhelm our immune system by doing that. So that's how the virus spreads around the body. It basically hijacks our entire machinery for building proteins to make its own proteins, and then essentially steals a bit of the cell membrane to package it all up, and off it goes to continue infecting things, which is pretty smart, particularly since the cells that it continues to infect are immune cells. So it is also taking out our immune system as it does so. Okay, here's another virus that is quite interesting. So this is Ebola. It's a really rare but deadly virus. It causes fever, body aches, and diarrhea. It's a hemorrhagic fever. So you bleed from all of your internal tissues and all of your every orifice. So your eyes, your ears, your nose, every hole in your body will bleed because it's hemorrhagic. You hemorrhage. It's horrible. So the virus spreads throughout the body, damages loads of tissues, organs, immune system, and causes blood clotting, cells to drop, and you end up with severe and uncontrollable bleeding. So it used to be called Ebola hemorrhagic fever because it causes hemorrhaging. It's now just called Ebola virus. It kills up to 90% of people who are infected. It's really hard to remove the bodies, to treat the patients without becoming infected as well. So it's full on hazardous biohazard suits to treat patients with this. It's not as contagious as respiratory viruses, believe it or not, because you have to be in contact with the blood and like body secretions. It is spread from people to people contact. So obviously caring for the sick is really key way of spreading this, and burial practices as well. It's really hard to dispose of the bodies of the dead from this virus carefully too. It can be spread from animal to animal through eating them, and again, monkeys, chimps, and bats, yet again, seem to be a very likely source of this. So it moves from people in essentially the same way. So you can't get it from the air though, or food or water. It's which is quite good. So usually, when I used to teach about this, when I first started teaching about this, I used to say there's no vaccine for Ebola, but there is now. So that's really good. There's no cure. Treatment basically includes giving a serum which destroys the infected cells and just managing the infection. You don't necessarily die, but it is really good. There you go. There's my notes. There is no vaccine to prevent Ebola. There is now though. So that's good news. It means we can probably eradicate it by vaccinating vulnerable communities, because it's very localized as well. It doesn't spread globally because it's not airborne. So the best way to avoid it is to stay away from regions where it's likely to develop. But obviously, if you live in one of those regions, it's not very easy. And you need to really avoid contact with animals that are potentially carrying it. So monkeys, chimps, gorillas, bats, always bats. And obviously healthcare workers have to wear an awful lot of PPE when they're working with people infected with that. So those are the sort of quite interesting ones, I suppose, viruses. There's loads of others, but those are the ones that I think a lot of people know about. So next pathogen on our list is bacteria. Here you can see a general bacterial cell, it has a cell wall, which we don't see in human cells. It's got a cell membrane, same as human cells, controls what enters and exits the cell. It's got cytoplasm, like human cells. It's where the watery sort of gel-like fluid where the reactions take place. But it also has these plasmids that I was talking about when I was talking about antibiotic resistance. They're extra little pieces of DNA you can see here. They're in a circular shape, often containing antibiotic resistance genes. The DNA itself, look, is not in a nucleus. It's just a big circle. Can't see it's a circle because it's all swiggled up. But if you straighten that out, it would be a big loop of DNA. It has flagella, not flagelli. I'm not sure why that says flagelli. Flagella, and these help it to swim a bit like a sperm cell. It also has cilia, which help it to move, very much like the cilia we were talking about in the respiratory system. These aren't shown on here. This one doesn't have cilia. These are pili. These are actually how two bacteria can attach to each other, and they can pass plasmids along these pili, thereby sharing antibiotic resistance genes. So here's an example of a bacterium that's not pathogenic. This is one that we use in food production. Lactobacillus bulgaricus is used in yogurt production. So they're not all bad. Some of them are good guys. So bacteria are prokaryotic organisms. Humans are eukaryotic. Plants and fungi are also eukaryotic. So eukaryotic means complex and prokaryotic is more simple. They are single-celled and many of them are disease-causing, but loads of them do really important things like breaking down dead materials, like dead plant materials, into turning it into soil, dead animals even. So loads of them have got really important decomposition roles. They reproduce through something called binary fission, and that just means one cell divides into two. So what happens is that big loop of DNA, the chromosomal loop, replicates. So you've got twice as much of it. All the little plasmids inside the cell also replicate. So cell will then just grow, as you can see on the screen. And then the membrane pinches inwards, it invaginates, we say, because this is called an invagination here. And it pinches inwards and it separates into two cells. So here is a good example. We have cholera. It's an infectious disease which gives you watery diarrhea. It can lead to dehydration and even death if it's untreated. You can get it by eating food or drinking water contaminated with bacterium. It's really common in places with poor sanitation that are crowded, often because of war or famine. It often occurs after natural disasters such as flooding or earthquakes, when people are living in sort of camps without running water. It's really dangerous for babies and young children. Symptoms of cholera can begin very quickly after infection, can be a couple of hours, but sometimes it can take up to 5 days. Sometimes symptoms are mild, but they can be very serious, and young children are very vulnerable. Essentially, this can all be avoided by making sure water supplies are clean. But obviously, in like disaster relief situations, it's not very, very easy to do. Very easy to do. So signs of dehydration, which are the main problem because of the diarrhea and vomiting, are rapid heart rate, loss of skin elasticity, dry mucous membranes, including the inside of the mouth, the nose, the eyelids, etc. It can lead to death pretty quickly. You can treat it with antibiotics because it's bacterial. So that's the good news about all of these bacterial diseases. They can be treated with antibiotics usually, bar the ones we were talking about yesterday. You can balance your body fluids by giving people drinks with sort of salts and sugars in, solutes and water. It helps them to hold on to their fluid, stop them becoming so dehydrated. You can hook people up to a drip as well. But if people do get too dehydrated, they can damage your kidneys and the brains and you can die. So all of this vomiting and diarrhea is the cause of this dehydration. So that is cholera. This is gonorrhea. So this is a sexually transmitted infection. this bacterium will grow in warm, moist areas of the body. So it likes to grow in the reproductive tract. It also grows quite nicely in the mouth, the throat, the eyes, the anus, anywhere really that it can get to and stay. So sometimes men with gonorrhea have no symptoms at all. So again, asymptomatic, you're much more likely to spread an asymptomatic disease because you don't know you have it. But symptoms, if you do get symptoms, will include a burning sensation when you're urinating, white or yellow or green discharge from the penis, and sometimes men get swollen or painful testicles. Women, symptoms are often mild, but they can have no symptoms. They can be non-specific because they might think that they have, I don't know, a bladder infection, which are quite common in women because the urinary tract in women is very small, obviously, because there's no penis, so it's very short. So urinary tract infections are quite common. So it's quite hard to identify as a sexually transmitted infection. But again, women can get painful or burning sensation when urinating and increased discharge from the vaginal area and also bleeding in between the monthly period where they'd normally expect to have bleeding. So it's quite easy to test for STIs, though, at the doctors. They are very routine if you go to the GP. So antibiotics can cure gonorrhea. There are, however, some drug-resistant strains in some parts of the world, including the United States. And that means, obviously, it's more difficult to treat because you haven't got antibiotics to treat it with. You can use the barrier method of contraception to prevent infection with gonorrhea. And here we have Mycobacterium tuberculosis, which we had a look at previously. This is another bacterial infection. It causes TB. It's a serious infectious disease which affects the lungs and can affect the brain and the bones as well. Whoops. So rod-shaped bacillus. So that's just the shape of the bacterium. It is non-motile. That means it can't move around, doesn't form spores. It requires oxygen to grow, and it's very, very slow growing. It's hard to treat with antibiotics because it has a waxy cell wall, so it's hard to get stuff into the bacteria to treat. So quite a nasty one. It's often referred to as the first diagnostic sign of AIDS. So when patients who have been infected with HIV develop AIDS, so their immune system is deficient, they often get TB as one of the first things. So the main reservoir for tuberculosis is humans. It is found in some wildlife like deer in the UK, and cows carry it. And the cows and deer can transfer it to badgers. And there's been a big badger cull in the UK because the farmers have been blaming badgers from spreading it to their cows, when, to be honest, the evidence suggests it's the other way around. The cows are spreading it to the wildlife. So there's been quite a lot of concern about TB in rural communities because of the effect on farming. So it can be spread via coughing and sneezing or speaking near to an infected person. So it can be spread relatively easily. So quite nasty. As we said, it's got this waxy outside, so it's quite hard to be destroyed, quite hard to destroy it inside the macrophages because of this waxy layer around the outside. And so it can avoid immune clearance by these phagocytosing cells. So primary TB, the initial infection may be asymptomatic or just cause mild symptoms. And then when you get latent TB infection, the bacteria are present in the body, but they just remain dormant. They may reactivate later. When you have active TB, it makes you very sick. You get a lot of weight loss, a very bad cough, fever, night sweats, and you cough up blood. You see it quite often in the movies with people coughing into a tissue. In the older days, if someone was going to signify they were going to die, that's what you'd see them cough into a tissue with blood, and it would be TB. Let me just check that there. I can see there's a question. Do viruses have motility? No, not really. They can just move around the body by getting into our tissues. And that's why when we sneeze and we project it, it's really good if they can make us sneeze or cough, because then we project the virus out a long way to infect someone else. So here is your example of a fungal infection. This is athlete's foot. So this is, it can be really unpleasant, actually. It's really easy to avoid, but you get it from essentially changing room floors is the usual route for this. Lives between the toes, causes dry skin, itching, discomfort. You have to have direct contact with spores or broken strands of the hyphae. Those are parts of the fungi. I used to swim a lot when I was a kid. I used to competitively swim. And what I would say is if you are swimming or around any pools ever, just we used to have pool shoes, like flip-flops basically, or which we just used to wear all through the change rooms anytime we were poolside, because obviously there's chlorine in the water, which essentially is like bleach. So it's pretty safe when you're in the water. But the standing water around the sides of the pool, you just shouldn't tread in it because it's full of, one, fecal bacteria, which is really disgusting, two, likely fungal infections, and three, viruses that cause things like verrucas. So, yeah, I wouldn't walk around wet changing rooms if you can help it. So you can reduce your likelihood of getting it by not just chucking your socks on after you're swimming, but really thoroughly drying your feet and then getting dressed. It says here fungal infections are easily treatable with fungal antifungal drugs, but they kind of are for some, like athlete's foot. But if it gets into your nails, the nail fungal infections are not easily treatable at all. Right, next group, protists or prototists. There's two different ways of explaining the name for this. Protists, I always called them, but exam boards increasingly call them prototists. This is the worst one that, well, just in terms of numbers, it's the one that causes malaria. So malaria is caused by a protist, a protozoan infection. These are essentially small single-celled organisms. They live in water or moist or wet places, most usually. You can see them under the microscope. Many of them are sort of free living. They live in soil and ditches or rivers or ponds. Some live as parasites in animals, and they are well known for a number of diseases, including malaria. Now, malaria is a mosquito-borne infectious disease caused by a protist from the genus Plasmodium. So another protist that causes disease is amoeba that causes amoebic dysentery. You might have heard of that one as well. So mosquitoes just spread this disease. They're the vector. They spread the disease. They're really widespread in tropical regions. Well, you get mosquitoes everywhere, but the disease, malaria, is really widespread in tropical regions because these flies, these mosquitoes, survive very well there. They can continually breed in the damp, wet environments that they like. So you can prevent infection by preventing being bitten. So mosquito nets are a good thing, and insecticide sprays can help. The disease itself actually results from the multiplication of the malarial parasites within red blood cells. Fever, shivering, joint pain, vomiting, very similar to flu, all of those symptoms. But you also get anemia, which is caused by hemolysis or hemolysis. And that's basically the red blood cells get damaged because they're full of these protocysts. So really, really not nice disease. You get night sweats, where you feel and you get really chilled, feel really cold, and you've got a fever. You can die from it. It's really nasty. So how this spreads is a mosquito infects a person by biting them and sucking their blood. They eat the blood, and then they leave the malarial parasites in the bloodstream of the person that they've bitten. In the liver cells, then the parasite, the protozoa, multiply. They change forms a few times as well. Eventually, they end up back in the bloodstream. The blood cells become infected. You lose a lot of those blood cells and end up with anemia. The parasite multiplies. The blood cells can burst and release the parasites into the blood. That person becomes bitten again by a new mosquito, and then they'll go off and bite someone else and transfer more parasites to the next person. It can be treated using drugs. So there are routes to avoid it spreading. I'm going to have to skip this, I think, guys, because we are running out of time a bit. And I need to tell you about these helminths, the worms. So these are parasitic worms that infect humans and animals. And there's three major groups that cause a lot of diseases, often through contaminated food, water, soil, or insect vectors, very commonly through water. So these are roundworms, the nematodes. They are cylindrical or round. They infect the intestines or other tissues. Here's a really good example. It causes a disease called elephantiasis. And again, this is spread by mosquitoes. Really horrible disease for the person infected. Tapeworms are flat segmented worms. Example is fish tapeworm. This comes from eating raw or undercooked freshwater fish. So this has got another water element to it. Mosquitoes need water to breed, so that's got another water element to it. So tropical regions, these diseases are really common. And then we've got the flukes. These are flat worms. They look a bit like a leaf. They're called leaf-shaped flatworms. They often require freshwater snails as intermediate hosts, which is really horrible. There's one called schistosomiasis. If you leave, if you go in the water and you've got cuts or like scratches on your feet, the flatworms can get into your body from there. And they need to spend a little bit of time in these snails to reproduce. It's part of their reproductive cycle, a bit like the plasmodium need to reproduce partly inside the mosquito, partly in humans. So if you can prevent these snails from, well, that's not the snail, that's the name of the disease, but if you can prevent these freshwater snails from gathering in the water, you can break the cycle of this horrible schistosomiasis disease from spreading. So that's another type of pathogens there. Okay, there you go. That is it. We got through it. So any questions, guys? I will see what I can do to answer. I know there was a question about HIV medication, which I'm not too sure about, to be honest. A question about PEP. It prevents getting HIV or passing it on. Why is it particularly dangerous to liver? Side effect? What does it do to liver? I don't know. Sorry, how that affects the liver. But many drugs, if they have side effects that affect the liver, it's because of the liver's detoxification role. I'm not sure if you remember when we were talking about the liver, we talked about it breaking down a lot of chemicals, alcohol, paracetamol, for example. I imagine it's to do with that. I imagine the liver breaks it down to get rid of it because essentially your body would recognize it as something that shouldn't really be in your body and would start breaking it down because it's a chemical that shouldn't really normally naturally be there. So I'd imagine that's what's going on there, to be honest, but I don't actually know. Any more questions on diseases? Yeah, I'd imagine, yeah, that's what you were thinking too. Yeah, probably is. It makes sense. That's how the liver gets damaged so easily with lots of different drugs and things like that. Okay, guys, if you've no questions, have a good rest of your day, and I will see you next week for homeostasis topic. Have a good weekend. Bye, everyone. Bye.